Today, we are looking at a set of source materials describing, well, what they call the nastiest antiviral concept on the planet.
And that is not an exaggeration.
No, it's not.
Yeah.
We are going to be dissecting the molecular armor plating known as the Queen-Kimi platform.
And look, that title is actually earned. This kind of deep dive is necessary because this strategy, it moves so far beyond just simple drug discovery.
Right.
This is an engineered biological system. It's designed from the ground up to preemptively defeat the three biggest failures we see in next-gen antivirals.
Which are what?
Rapid viral escape through mutation, the huge problem of needing impossibly high therapeutic doses, and maybe most importantly, patient compliance.
Exactly.
Yeah.
So our mission today is to put ourselves through the same self-critique the engineers did. They called it the reality check.
Where they just tore their own ideas apart.
They did. And then we'll look at the high-tech fixes they came up with using some pretty deep chemical and molecular engineering. If you understand the problems, the fixes are just brilliant.
Yeah, if you zoom out, Queen Kimi is so impressive because it solves these problems all at once, simultaneously. We're looking at a three-pronged system.
Okay, lay them out for us.
You have the advanced entry shield decoy, an automated genome shredder, and then a totally redundant universal safety system. The key insight is that the engineering had to be multi-layered.
Because single solutions just don't work.
They always fail against biological complexity. Always.
Okay, let's unpack this, starting with that first line of defense, the entry shield. The concept seems straightforward enough. It's an engineered ACE2 FC decoy.
Right, so it's designed to act like a fake receptor, basically.
To bind up the viral spike protein before it can ever infect a cell. Sounds like a solid start.
Yeah.
So what was the first reality check they ran into?
The first reality check was evolution, pure evolutionary pressure. Their own data showed that these simple, single-point decoys, they're actually evolution accelerator.
Accelerated. You mean they make the problem worse?
Yes. In their mouse models, the SARS-CoV-2 virus, specifically the RBD, managed to escape these decoys in less than 2 weeks.
Wow.
You're effectively giving the virus a training course on how to mutate faster.
So the single binding point was just too easy for the virus to work around. But that wasn't the only problem, right? They also had physical and chemical flaws.
That's right, huge ones. The affinities were just too low to be effective. The first Velcro peptides they tried had micromolar KD, very weak, and physically they were unstable.
What do you mean by unstable?
The FTAC part would swivel at body temperature at 37 Celsius, and that exposed these hydrophobic patches.
Which means they all clump together.
Exactly. They'd aggregate and just fall out of solution. Useless.
And even if you ignore all of that, you still hit the brick wall of concentration.
The dosing problem was immense. I mean, they calculated that a standard 30 microgram mRNA dose, it only gives you a peak serum concentration of about 50 nanograms per milliliter.
Which is nearly enough.
Not even close. For real neutralization in vivo, you need at least 1 microgram per milliliter. That's a 20 to 30-fold gap. A 2 to 3 log shortfall right out of the gate.
So you have structural weakness, aggregation, evolutionary pressure, and you can't even get enough of it into the body. That's a full-scale catastrophe for an early drug concept.
It's a complete non-starter.
Okay, so let's get to the fixes.
Yep.
How did they engineer the decoy to actually beat viral escape?
They used a triple solution. First, to fix the evolution problem, they made the decoy bispecific.
Bispecific meaning it binds to two things at once.
Exactly. They kept the trimeric ACE2FC structure, but they chemically grafted a second, highly specific binder onto it. And the second binder, it locks down the open HR1 groove.
That's a vulnerable part of the virus's fusion machinery.
A really vulnerable part. So instead of the virus needing one random mutation to escape,
It now needs two successful mutations at the same time, on two totally different parts of its spike protein. It's a molecular trick shot.
it's a trick shot. And the math on that is just brutal for the virus. The probability product for a simultaneous escape dropped to about 10 to the minus 8 per genome copy.
Okay, put that in perspective for us. 10 to the minus 8.
Look, it's like needing to win a 1 in 100 million lottery. The virus is trapped.
So the evolutionary trap is solved. That's a
Yeah.
huge step. But what about the physical problems? The low affinity, the aggregation? That sounds like a different kind of engineering.
A totally different approach. It required synthetic chemistry. They had to replace that simple peptide Velcro completely.
With what?
They turned to a lab to synthesize a trivalent silane LN3 dendron.
The dendron. For anyone not familiar, that's like a tiny branched tree-like molecule, right?
That's a perfect way to describe it. It's tiny, only 2.1 kilodaltons, but its shape is everything. The symmetry is the key.
And why does that symmetry matter so much here?
Because that symmetry is what gives you stability and, more importantly, high avidity. That means you get incredibly strong binding because you have multiple attachment points at once.
So the affinity goes up.
It doesn't just go up, it skyrockets. It went from that weak micromolar KD to less than 10 picomolar.
Wait, micromolar to picomolar. Can you give us a sense of scale? How much stronger is that bond?
It's exponentially stronger. A picomolar bond is roughly 100,000 times tighter. It's the difference between Scotch tape and a weld.
Wow.
And because that dendron structure is hydrophilic, it physically masked those hydrophobic patches. The result, zero aggregation, even when they stressed it at 55 degrees for 2 weeks.
So it completely fixed the stability problem.
Completely.
Okay, so the decoy is now chemically stable and evolutionarily bulletproof. But we still have that fundamental delivery problem. We need 20 times the serum concentration.
Right, we've perfected the molecule. Now we need to mass produce it inside the patient's body, and that's where the second major fix came in.
Self-amplifying RNA.
Self-amplifying RNA, or SARNO.
Which, for the listener, is RNA that basically makes copies of itself once it gets into your cells, right?
That's it. It carries the instructions for the decoy and the machinery to replicate those instructions hundreds of times over. So they switch to a higher dose, 100 micrograms of SARNA, delivered in a very specific lipid nanoparticle.
And LNP, I saw it was rich in DSPC. Why that specific lipid?
That was the key to steering it. The DSPC-rich formulation acted like a molecular GPS, and it reduced liver tropism, the tendency for it to all just pile up in the liver by threefold.
Which is a big safety improvement.
Huge. But the major win was the concentration. In cynomolgus monkeys, this strategy got the serum decoy levels to 3 to 5 micrograms per milliliter and kept it there for 10 days.
And that's the sweet spot. That's the concentration they needed. And the 10-day window is just enough time for the second layer of the strategy to kick in.
The genome shredder.
Let's move to that. The Cas13 chop lock system. This is the cleanup crew.
It's the cleanup crew, designed to go into infected cells and just chew up the viral RNA genome. But again, the reality check was brutal. Massive bottlenecks.
Starting with manufacturing scale. Their initial design needed 2,000 different guide RNAs to cover all the variants.
It sounds comprehensive, but that translated to 4 milligrams of RNA mass per dose.
And that mass creates an impossible manufacturing limit. I mean, if your GMP max batch size is 2 grams, which is pretty high.
You can only make 500 doses per batch. In a pandemic, it's unacceptable, a non-starter.
So they couldn't just make more RNA, they had to shrink the need for RNA.
Exactly. And the fix was pure bioinformatics. They ran 50,000 full genome alignments and used something called Shannon entropy analysis.
So instead of trying to cover everything, they looked for what?
They looked for hyperconserved 20mers, sequences with extremely low Shannon entropy, which means they barely ever change across any of the variants.
So you don't need 2,000 guides hitting everything. You just need 200 guides hitting the most critical, unchanging parts of the viral genome.
That's it. And that dropped the RNA mass tenfold, down to 400 micrograms total. Problem solved.
Right.
But then came what they called the compliance black hole.
Which was tied to the safety switch. The original plan used a daily drug, AP1903. Why is relying on the patient for a daily safety switch such risk?
It's a regulatory nightmare. We just know from decades of pharmacology that compliance for drugs that need daily action, it rarely lasts more than 7 days effectively.
The compliance black hole.
Right. And if your Cas13 might stick around longer than needed, relying on a patient to take a pill to turn it off is just asking for trouble. High risk of patient error.
So they needed to automate safety, take the human element out. And this, to me, is the most elegant piece of engineering in the whole thing.
The auto-off ribozyme switch. It is the major breakthrough.
Tell us how it works.
So they replaced that manual pill with a 3' embedded hammerhead ribozyme, an HHRS, and it's built right into the Cas13 mRNA itself.
And the trigger for this self-destruction isn't another drug, it's the host's own biology.
That is the genius of it. The ribozyme is engineered to self-cleave to destroy the mRNA only when a specific microRNA in the liver, called miR-122, rises above a certain baseline.
And how does that specific microRNA, miR-122, correlate with recovery from the virus?
It correlates perfectly. When you have a high viral load, the liver is under massive stress, and miR-122 levels are suppressed. As the infection clears and the viremia falls, the liver recovers, and its natural gene expression, including miR-122, climbs back up.
So the rising miR-122 is the body's own natural all-clear signal.
It is the automatic all clear.
So the drug senses the body is winning, and then it immediately starts to dissolve itself.
That's exactly what happens. It triggers the Cas13 mRNA to self-destruct and fast. In their models, the protein's half-life dropped from 14 hours to just 45 minutes once that threshold was crossed.
No extra pill, no patient action required.
Nothing. The drug just cleans itself up once its job is done.
That is the ultimate solution to the compliance black hole. It makes safety automatic.
It's what makes it deployable at scale. And
Yeah.
they had to apply the same check and fix process to everything else. I mean, even the panenzyme inhibitor, the FN3 monobody, got a huge overhaul.
Right. They turned that into a bispecific agent as well.
Yes. They linked two FN3 loops. Loop 1 binds the viral engine, the RDRP. Loop 2 hits the protease, 3CL-Pro. By hitting two critical targets at once, the potency went up 100-fold.
100-fold. That means a huge reduction in the amount of drug you actually need.
Absolutely. The clinical dose dropped from 20 milligrams per kilogram down to a much more manageable 2. It was just optimization at every single level.
But what about the ultimate safeguard? I mean, if despite all these automated systems, a patient has a severe adverse event, they still need a kill switch.
A universal kill switch.
Yeah.
And they built one. This is the final layer of safety. They encoded a unique drosha-cleavable hairpin sequence into every single therapeutic mRNA in the platform.
Okay, so drosha is a cellular enzyme that edits RNA. How do they leverage that to shut the whole thing down?
If there's a serious adverse event, the patient gets a single tiny IV dose of insurinase that targets DGCR8.
And DGCR8 is essential for drosha to work.
Right. So you inhibit GGCR8 and drosha activity plummets by 90% within 30 minutes. Then all those therapeutic mRNAs with their special hairpin get diced up and destroyed. Protein synthesis just stops.
30 minutes. That's an immediate comprehensive shutdown button for the entire system.
It's the kind of safety profile that gets you past regulators.
So let's talk about the final metric. Cost. All this incredible engineering must come at a price.
You'd think so, but the optimizations were successful on the financial side too. At a billion-dose scale, the total projected cost per full course is about $25.
$25 for this whole multi-layered system.
Yep. Our sources confirm that's well under Paxlovid's $40 to $70 cost and way cheaper than monoclonal antibody cocktails. The big components are all under $8 each.
And they hit their success goals, proving it was effective and safe, not just cheap.
Absolutely. The non-negotiable gates were things like Cas13 had to reduce viral RNA by at least 2 logs in 24 hours. And critically, all therapeutic mRNA had to be gone below qPCR detection by day 10.
Which proved that the miR-122 suicide switch actually worked in a living system.
It validated the whole automated safety concept.
So what's the big takeaway for us, for the listener? The Queen Chemi strategy shows that the next generation of therapeutics won't be about just finding one potent compound.
No, it's about fully integrated systems. Systems designed to beat logistics, evolution, compliance, and safety all at the same time while staying affordable.
The conclusion in the source material is pretty provocative.
It is. It says, ready to make some viruses wish they'd never evolved. This deep dive really shows that maximizing potency, like that 100x gain, is only valuable if you couple it with a fail-safe.
Right.
The true innovation here is the automation of safety, taking human error and high cost out of the equation. That's the game changer.
A perfect summary. The automation of safety. So for you, the listener, we'll leave you with this question. Which of these molecular fixes, the trivalent dendrin that created hyperstability, the SARNA dose boost that solved delivery, or that automated miR-122 kill switch, which one do you think had the biggest single impact on taking this from a lab concept to a real pandemic-scale therapeutic? We'll catch you next time for the next deep dive.